People are furiously debating right now whether ivermectin is a miracle drug for COVID-19 or whether it's completely worthless. So far, we've only had low-quality studies to draw from, along with plenty of strong rhetoric on both sides. Now, however, we have two studies, meta-analyses of all the studies done to date, a process that may give us a more reliable picture. There's only one problem. They come to completely opposite conclusions. What's going on? Let's have a look. Before we begin, the necessary disclaimer. 1. This video is a description of recent evidence and debate around ivermectin. It does not advocate for or against it. 2. You should never ever take medical advice from YouTubers, even those that claim medical credentials for themselves, which I definitely do not. Nothing in this video constitutes medical advice of any sort. It's an analysis of a public policy debate. And by the way, yes, YouTube has restrictive community guideline policies in this area, but those caveats would not change even if they didn't. Right, now we've got the small print out of the way, let's quickly recap the debate over ivermectin and why it's important. Global pandemic, big impacts across the world, both from the health impact of the disease and the disruption of the attempts to contain it. Effective vaccines, of course, but limited supplies of those, even at the astonishing scale of production that has been achieved. Poorer countries particularly hard hit, where low-cost, easily available and effective drugs would be extremely useful. But as always, we want any such drugs to be fully studied and shown to be effective and safe. The global research community has failed to do a timely large-scale clinical study of ivermectin following some initial positive signs from smaller studies. That failure has led to a growing impatience and cynicism, with advocate groups jumping up, internet exaggerations and rumours, and all of the rest of it. That has often led to those low-quality small studies being given the weight of certain proof. But professionals in this area attest to their experience that early signs of promise in such studies routinely turn into disappointment when proper studies are done. Now, a number of large-scale trials have rather belatedly been started, but into this situation now has come two other studies, what are called meta-analyses. A meta-analysis looks at various small studies that have been done, aggregates them to find a more reliable picture of their findings. Individual low-quality studies may suffer from a point of bias introduced by poor sampling, faulty process, or so on. So carrying out a meta-analysis of all the studies makes it likely that different biases will cancel each other out, and the more reliable signal will shine through all the noise. The thing is, one meta-analysis, Bryant et al., finds very conclusively that ivermectin is a highly effective treatment for COVID-19. The other one, Roman et al., finds very conclusively the exact opposite, that ivermectin is of no value at all and should not be used. Both studies purport to use more or less identical processes, so any ordinary observer who's just interested in what's true might well find their head exploding at this point. How can two studies drawing from the same universe of source material, using the same methods, come to such different conclusions? Now, of course, this is an area where there are some passionate advocates. As far as they're concerned, it's just that the other lot made grievous errors. And maybe they're right, but for the rest of us, it forces us to look at the detail, see what the experts are debating and arguing about. Because, let me make my standard disclaimer here, I am not an expert in this field. I look at the published research, I report what it says. When scientists argue amongst themselves about that research, I can look at the substance of their debate. If the principles are clear, I might be able to take a view. Sometimes it's entirely possible to do that. If they are more technical in nature, then I can report the spirit of what they're saying and watch with interest for how it resolves. In that situation, many non-experts who leap to one side or the other are simply expressing their prior preference. Well, I would try not to do that. But in any case, I don't even have a prior preference. In this case, I'm not sure why evaluating a drug attains the same dynamic as supporting a football team, but for some people it seems to do that. So with that said, let's have a look at the detail here. The difference between the studies seems predominantly to come from how the studies that you're reviewing, how those are evaluated. There's a process for deciding what the quality of those studies are that you're reviewing, upgrading their value in the results or downgrading them accordingly, and particularly grading them for risk of bias. So both meta-analyses make an evaluation of the robustness of the studies they cover, and it's here where the differences come in. The first meta-analysis, Bryant et al., carried out by Tess Lowry, gives the detail behind its assessment of the various constituent studies. Some are rated as having high risk of bias, some very low, a number of others with high or low, but with some areas of uncertainty because of gaps in the information about the process. Generally, critics agreed that the methodology overall used by the paper was good. Criticism seemed to come in two areas. One is that the paper states there is no evidence of publication bias. Now, publication bias comes because journals are more likely to publish studies that demonstrate a thing than they are to publish one demonstrating the absence of a thing. It's a well-known issue with research publishing. In reality, studies that show that something doesn't work should be valued just as highly because they're still giving you important information, but they're not. How can you tell if there's publication bias affecting the studies that you're looking at, for this meta-analysis? Well, one way of doing it involves a funnel plot, where the Lowry paper produces and declares that it did not suggest any evidence of publication bias. But some researchers have looked at that plot and said, well, actually, it kinda does. The principle of a funnel plot is that the studies of high precision, higher up the y-axis, will tend to plot near the average, and studies with low precision will spread evenly on both sides of the average, creating a roughly tunnel shape. This is a generic example of a plot you would expect in the absence of publication bias. Now, it's certainly true that the funnel plot in the study sees the low precision studies somewhat unevenly distributed. So that is a little suggestive, but the more substantive criticisms come in the bias rating of the various papers. So Gideon Meyerowitz-Katz wrote about this on Medium and was also interviewed for the BBC's More or Less programme. He reviewed the bias rating and said this: the authors used Cochrane's risk of bias tool to rate studies in the review and produced the below table of the results. Some of these studies are definitely fine. For example, the Mahmood study was extremely well conducted and it's hard to find fault with the author's judgment. However, there were a few studies for which the rating seemed a bit off, although it's worth bearing in mind these ratings are somewhat subjective regardless of the context, i.e. I could be wrong too. In particular, if we look at the Naei 2020 and Algazar 2020 studies, the ratings seem a bit optimistic. For example, Naei is a randomised trial that recruited people who were PCR negative for COVID-19, as well as those who tested positive. But they somehow ended up with a nearly 50% PCR negative in the control group, only 20% negative in the intervention. This is an issue for both the randomisation and the allocation concealment elements of the study. But both of these are rated as low risk of bias, which doesn't seem to take this issue into account. The Algazar study simply has no information whatsoever on allocation concealment at all, and the two sentences on randomization procedures actually contradict each other, yet it's still rated as low risk of bias for both of these studies. The thing is that this makes a big difference to the outcomes. He found that if you excluded those two studies from the group and re-ran the analysis, the results are entirely reversed. A massive benefit from ivermectin becomes no benefit at all. But he added, even if you didn't agree with those alternative assessments, if you took only the 3 studies that the authors of the meta-analysis themselves considered to be the lowest risk of bias, in other words, the highest quality studies, and you only included those ones, then again you failed to find any benefit for ivermectin. If that analysis is correct, then at the very least you have to conclude that the high degree of confidence of the authors in their findings, well, seems to be a bit misplaced. Because nothing in that states that ivermectin doesn't work, just that the evidence is low quality. And you're making subjective decisions about which studies are stronger than others, and depending where you fall on that judgment, you come up with different results. Now, Tess Lowry and one of the other authors, Edmund Fordham, were not entirely thrilled with the case made on the BBC More or Less programme, which was basically the same as the one that I've just described. They produced a written rebuttal, headed with the fairly uncompromising headline, "Attempting to Discredit Landmark British Ivermectin Study," and drove the point home with the subheading, "Medical Disinformation from the BBC." So, what were the counterpoints? First, they complained loudly that none of the authors of the study had been contacted for comment, which is fair enough. I mean, the BBC programme was actually really quite short, just about 9 minutes or so. Even so, you should have been able to get some sort of response comment in there. They complained that the programme referred to the other meta-analysis, which we'll get to in a moment. They talked about why that one's inferior, shouldn't be held alongside their own. And then they said this: Meyerowitz-Katz attempts to discredit Bryant et al. On the basis of risks of bias and certainty of evidence, but failed to explain that this would not change any of the numerical estimates of the magnitude of the effect, e.g. on mortality, on clinical improvement, or prevention of infection, etc. First, I didn't get the sense that Meyerowitz-Katz was, quote, attempting to discredit their paper. I thought it was a fairly focused analysis that concluded the paper might or might not be right, not that it was definitely wrong. I also thought that he specifically said that it did change the result. His explanation for how it did that seemed straightforward and understandable, and also specific as to the papers that he was referring to. Laurie and Fordham continued, whilst assessments of evidence certainty do involve subjective aspects, they are all subject to strict criteria and should be carried out independently by two reviewers. Bryant et al. Observed those criteria. Whatever process was used by Roman et al., the result was only multiple instances of data misreporting. So from what I can see, they acknowledge that there are subjective aspects of judgment. There are processes to minimise the risk of error, and they followed those processes fine. It doesn't necessarily respond to the specific point that was being made, and then they quickly default back to an attack on the other paper. Harford and Meyerowitz-Katz between them also failed to explain that in the context of a systematic review, phrases such as moderate certainty or very low certainty have a precise technical meaning. These concern overall confidence in the magnitude of effects and whether further research is expected to change the quantitative estimate. They do not refer to certainty about whether an effect exists at all. To my eyes, I don't get the sense that the rebuttal goes to the heart of the points that were being made, but they then go further. The effectiveness of ivermectin in the prevention and treatment of COVID-19 is not ultimately shown by meta-analyses of randomized clinical trials. It is now glaringly obvious from multiple observational trials and case studies from whole countries or states within them, showing how ivermectin use crushes both case and death statistics. If you think that last paragraph seems a bit on the polemical side, I would have to agree with you. The studies they refer to here provide nothing like the degree of proof that is claimed in that paragraph. For instance, there's this study, which I discussed in my previous video on ivermectin. It shows a correlation between African countries that gave ivermectin as a prophylactic treatment to citizens against river blindness and low levels of COVID cases and mortality versus those that didn't. But even the authors of the study agreed that this was only evidence of a correlation, not necessarily of a causation. Jumping to conclusions from whole nation effects is something you should do with extreme caution, because there are so many potential variables in play. Plus, if you revisit that graph, some of the countries that were in the happy group, ivermectin dosed and with low incidences of COVID, well, they've seen some changes, such as Uganda, which has seen a new spike in cases and deaths, and Mozambique likewise. These are at much lower levels than Europe, America and Asia, but then levels for nearly all African countries are at much lower levels, and ivermectin doesn't provide the explanation for that. Limited amounts of testing and reporting seems to be at least part of the explanation. Poor quality data means that at the very least you should be careful of your conclusions. So if you're going to represent that paper with the hubristic language of crushes both case and death statistics, I'm going to treat your representation of the facts with some caution, because you're talking like a campaigner, not like a scientist. But what about that other paper, Roman et al.? Is that the study done right, without the same potential biases and therefore a more reliable conclusion? Or is it similarly problematic, or is it even worse? Well, first thing to note is that it covers some of the same papers, but not all. Indeed, where Bryant et al. Covered 24 studies with 3,406 participants, Roman et al. Had 10 studies with 1,173 participants. Now, if all the omitted studies were counted out because of low quality, that could be justifiable. It could well make their paper more accurate a reflection. But as we've already seen, the selection and rating process can follow recommended procedures, but still remain somewhat subjective. That being the case, it does seem rather bold for the study to conclude as it does. Ivermectin is not a viable option to treat COVID-19 patients. That seems an uncompromisingly certain statement for an evaluation of a subset of lower quality studies. And the robustness of the work has been roundly attacked by the other side. The most telling of the attacks is directed at the existence in the first preprint of errors relating to the results of some of the studies. In particular, Nye et al., the study that Mayo-Witz-Katz singled out as one of the two with the oddly forgiving bias assessment in the other paper, the Roman et al. First draft inverted the results for that paper, putting the ivermectin results in the control column and vice versa. The critics complained that although the mistake was corrected after it had been pointed out, that didn't seem to have altered the conclusions in any way. And they also alleged that some other errors remained. That paper is currently accepted for publication but not yet peer-reviewed, so whether those errors will be ironed out and whether it will be finally published is an open question. Nevertheless, it's hard not to conclude that both of these papers are inconclusive at best, and the nature of the argument simply highlights that large-scale double-blind studies are needed to resolve the question once and for all, like one that has just been started with Oxford University. Not everyone agrees. The Ivermectin campaign has sent a letter to Oxford University calling its proposed clinical trial highly unethical. They suggested the evidence was already so overwhelming that having a control group of people who didn't receive ivermectin was against everything we swore an oath to as doctors. Which is not wholly persuasive when ivermectin is currently not authorised for treatment and a robust large-scale trial would be the vehicle for changing that. They also suggested that the design of the study would be unlikely to provide reliable evidence and said it was because it was open to people who had been showing COVID-19 symptoms for 14 days or less, arguing that the virus naturally clears the body in 5 to 7 days. But presumably a good proportion of people who joined will be in those early stages. That will all be recorded. But if someone is still feeling symptoms a number of days later, then presumably the virus has not cleared the body at that point. In any case, a well-run trial like this will record all the factors, will follow all the quality procedures, so attacking it in advance seems to be a little as though people are not wholly confident of their ground. And the critics of ivermectin do have at least some persuasive arguments, although note persuasive doesn't necessarily mean proven. Yuri Dagin, who has been in an intense and not entirely good-humoured exchange with ivermectin advocate Brett Weinstein, pointed out that if you look to studies that have followed the best process, the randomised placebo-controlled double-blind format, then it comes back with ivermectin rated as either ineffective or very mildly effective. So he pointed to papers including this one, Vallejos et al. From Argentina, which concluded ivermectin had no significant effect on preventing hospitalization of patients with COVID-19. Patients who received ivermectin required invasive MFS earlier in their treatment. And this one, Abd El Salam et al., which said, although there was no statistically significant difference in any endpoints by ivermectin doses, 12 micrograms per day for 3 days, there was an observed trend to reducing hospital stay in the ivermectin treated group. And this one, Lopez Medina et al., concluding, among adults with mild COVID-19, a 5-day course of ivermectin compared with placebo did not significantly improve the time to resolution of symptoms. The findings do not support the use of ivermectin for treatment of mild COVID-19, although larger trials might be needed to understand effects on other clinically relevant outcomes. If ivermectin was as highly effective as the advocates portray, you'd expect more consistency in results and that the higher rated trials in terms of lack of bias would be the ones to show the most effect. But nevertheless, it's all noise. We need the larger trials to cut through it. I'm not qualified to judge the scientific merits or otherwise of any of the studies. I've reported some of the arguments about them from other people, but I am pretty happy to call BS on people arguing that anything about this situation means that we don't need any more information. And that view has been rather confirmed in the last few days with the publication of, yes, another meta-analysis, this one by Hill et al. Like London buses, meta-analyses seem to arrive in threes. This one covered studies that were available up to the 12th of May, and it was 24 randomised controlled trials constituting a total of 3,328 participants. And it rather confirms the point that ratings of bias of papers is subjective, because it was different to the others on several measures. Meyerowitz-Katz had said that 2 of the studies in the first paper had been rated low bias but were more dubious. The Hill et al. Paper agrees with him on one of those, El-Ghazar 2020, but not on the other, Naei 2020, which it rates as good. The one that Meyerowitz-Katz had specifically agreed with the authors of the first study, saying that he agreed that its methodology was good, Mahmoud et al. This study rates as more suspect. This meta-analysis finds that there were some positive observed effects from ivermectin. I quote, "This systematic review and meta-analysis of 24 RCTs showed ivermectin treatment reduces inflammatory markers, achieves viral clearance more quickly, and improves survival compared with standard of care. The effects of ivermectin on viral clearance were stronger for higher doses and longer durations of treatment. These effects were seen across a wide range of RCTs conducted in several different countries." But the authors then went on to highly qualify the results by noticing the significant inconsistencies in all of the various studies. A key limitation to this meta-analysis is the comparability of the data, with studies differing in dosage, treatment duration, and inclusion criteria. Furthermore, the standard of care used in the control arm differed between trials. In this meta-analysis, trials that used active controls such as hydroxychloroquine or lopinavir-ritonavir were combined together with those that used placebo or standard care. However, lopinavir-ritonavir and hydroxychloroquine have shown no overall benefit or harm in large randomized trials and meta-analyses. Because obviously if you give one group ivermectin and you give the control group a different care regime that is actually harmful to them, then the comparison of symptoms between the two is kind of hard to make, because it could be down to either of those two factors. And it concluded, therefore, that the need was reinforced for larger trials. Several other repurposed medications have shown promise in early, smaller trials. For example, rofosbuvir, daclatasvir, colchicine, and remdesivir. But the benefit was not seen in later, larger trials. This meta-analysis of 24 RCTs in 3,328 patients showed a 56% improvement in survival, faster time to clinical recovery, and signs of a dose-dependent effect of viral clearance for patients given ivermectin versus control treatment. This benefit needs to be validated in larger confirmatory trials. So a study that is clear-sighted about the quality of its component studies, clear about potential benefit, but also the limits to the degree of proof, calling for qualitative studies to answer the question. I'm inclined to trust that outcome more than I am the ones that brook no uncertainty with their wildly contrasting positions. As Gideon Meyerowitz-Katz said on the BBC programme, it's an indictment on the research community that such trials weren't done long ago, given that so many countries are, in desperation, already widely prescribing ivermectin. Now that is now being rectified, not just by Oxford University, but several different studies. Let's see what comes back and keep an open mind without prejudice. Because it would be great news if a commonly available and cheap drug was found to be genuinely helpful. And the fact of its attempted suppression by some folks YouTube would give folks like me something to talk about for many months. But if it's not helpful, the people currently taking it, thinking it's really helping them, they need to know that it's not, and that they need something else. As Richard Feynman said, for a successful technology, reality must take precedence over public relations, for nature cannot be fooled. thanks for watching this video. If you liked it, please share with anyone else you think would also enjoy it. Word of mouth is really important to us. And if you've not subscribed yet, what are you waiting for? As the saying goes, that subscribe button won't smash itself.